Coming to another way in which glucose is oxidized is the glucuronic acid pathway or uronic acid pathway. So we have seen the major way in which glucose is oxidized is glycolysis, and apart from that, we have another way in which glucose is oxidized is HMP pathway, and the minor route in which glucose is oxidized is uronic acid pathway, also called as glucuronic acid pathway. So this pathway is not concerned with the generation of energy, but it produces an important compound called as UDP glucuronic acid. So that is the significance of this pathway in humans. Whereas in case of lower animals, this glucuronic acid pathway can be utilized for the synthesis of vitamin C. Ascorbic acid can be synthesized from this pathway, that is uronic acid pathway. So that is the significance of this pathway. Coming to reactions of uronic acid pathway or glucuronic acid pathway, here UDP glucose, which is the active form of glucose, is converted into UDP glucuronic acid by the action of enzyme called as UDP glucose dehydrogenase. Right. So UDP glucuronic acid in the next reaction by the action of enzyme called as UDP glucuronidase. So that glucuronidase removes the UDP and it forms D-glucuronic acid. Right? And what happens to that D-glucuronic acid? That D-glucuronic acid is now converted into glucuronic acid by the action of enzyme called as reductase. So that glucuronate reductase converts glucuronate, D-glucuronic acid or D-glucuronate into L-gluconic acid. So D isomer is converted to L form, and that is L-gluconic acid. And this L-gluconic acid has two phases. In case of lower animals, L-gluconic acid is converted into ascorbic acid by the action of enzyme called as gulonolactone oxidase. So that particular enzyme is absent in humans, whereas it is present in the lower animals. So that's why they can synthesize the vitamin C, whereas we cannot synthesize vitamin C because of defective enzyme called as gulonolactone oxidase. Now, in case of humans, the fate of L-gluconic acid is it is converted into a pentose sugar called as xylulose 5-phosphate. So in this conversion of L-gluconic acid to D-xylulose involves some set of reactions where first gluconic acid is converted into L-xylulose. So there is oxidation and decarboxylation occurs, so that L-gluconic acid is converted to L-xylulose. So L-xylulose is converted to xylitol by the action of enzyme called as xylitol dehydrogenase. So whenever there is deficiency of this particular enzyme, xylitol dehydrogenase is there, that leads to a condition called as essential pentosuria. So there is increased excretion of xylulose in the urine, that is pentose sugar. So presence of pentose sugar in urine, we call it as pentosuria. Yeah. So that is the significance of that particular enzyme, xylitol dehydrogenase. So this xylulose in the urine can be detected by Benedict test, because Benedict test gives the positive test when you react when it reacts with the xylulose in the urine. Right. So that xylitol, which is formed by the action of enzyme xylitol dehydrogenase, is converted into D-xylulose by the action of enzyme called as xylulose reductase. So that D-xylulose is converted into D-xylulose 5-phosphate and by the action of enzyme kinase. So xylulose 5-phosphate enters into the pentose phosphate pathway, and there by the action of enzyme transcriptase interconversion of ribose 5-phosphate xylulose 5-phosphate occurs. So this is how UDP glucose, which enters into the uronic acid pathway, is finally converted into D-xylulose 5-phosphate. And here the important compound which is produced in this pathway is D-glucuronic acid. So this is very important part of the uronic acid, because coming to significance, we will see under significance what is the significance of this uronic acid pathway is it produces UDP glucuronic acid. That UDP glucuronic acid is utilized in many reactions in our body. For example, it acts as the conjugating agent. Right? Bilirubin, which is produced in the heme catabolism, is detoxified. It is converted to soluble form when it binds with the glucuronic acid. The source for that glucuronic acid, the active form of glucuronic acid is UDP glucuronic acid. So that glucuronic acid which is required in the conjugation of bilirubin is supplied by this uronic acid pathway. So conjugation of bilirubin is the most important reaction where glucuronic acid is required, right? And the other reactions where glucuronic acid is required is in other detoxification reactions. So in conjugation, phase 2 of detoxification, that is conjugation reactions, the most important conjugating agent is glucuronic acid. So whenever it combines with a compound, it increases the solubility of that toxic compound so that it can be easily excreted from the body. So that is the significance of conjugation, and the most important conjugating agent is glucuronic acid, and the active form of glucuronic acid is UDP glucuronic acid. Apart from that, this UDP glucuronic acid is also involved in the conjugation of various drugs. It is involved in the conjugation of various steroids. It is used in the synthesis of glycosaminoglycans. So for the synthesis of some important heteropolysaccharides, we require this glucuronic acid. So these are the important functions of glucuronic acid, which is produced in the uronic acid pathway. So this is the significance in case of humans, whereas when you see significance in case of lower animals, this glucuronic acid, uronic acid pathway is concerned with the synthesis of ascorbic acid. Now, so we have seen what is essential pentasuria. So whenever there is defective xylitol dehydrogenase is there, that leads to increased excretion of pentose sugar xylulose in the urine, that is called as essential pentasuria. Now coming to metabolism of galactose. So galactose is metabolized in our body, and the most important enzyme is galactose 1-phosphate uridyl transferase. It has a clinical significance. Whenever there is deficiency in this particular enzyme, it leads to a condition called as galactosemia. So for the conversion of galactose to galactose 1-phosphate, we require galactokinase. So after that, galactose 1-phosphate is converted into glucose 1-phosphate and UDP galactose. That conversion requires galactose 1-phosphate uridyl transferase. So galactose 1-phosphate uridyl transferase deficiency leads to a condition called as galactosemia. So what are the features of galactosemia? Whenever there is deficiency of this particular enzyme, it leads to increased concentration of galactose in the blood. So that's why we call it as galactosemia. And the most important features of galactosemia are there is mental retardation, there are eye manifestations like there is cataract seen in case of galactosemia. Apart from that, there is jaundice, there is increased accumulation of galactose in the liver because it is metabolized in the liver. So that leads to hepatomegaly, enlargement of liver occurs, and there is increased, as I told you, there is increased concentration of galactose in the blood, and that leads to excretion of that galactose through urine, and presence of galactose in urine is called as galactosuria. So galactosemia, galactosuria, and cataract, mental retardation, hepatomegaly. So all these are the important features of galactosemia, which is because of deficiency of enzyme called as galactose 1-phosphate uridyl transferase. So this galactose can be utilized in the synthesis of milk, because the composition of lactose is glucose plus galactose. So mammary glands, they synthesize milk, and the source of galactose is, so whatever glucose we will take, that can be converted into galactose, and galactose can be converted into glucose by the action of enzyme called as epimerase. So the function of galactose is it can be used for the synthesis of, one of the functions is it can be used for the synthesis of milk, and the other function is this galactose can be used in the synthesis of some heteropolysaccharides. So this is the significance of galactose metabolism, and the clinical condition associated with the galactose metabolism is galactosemia. Thank you.